Okay. Hey, Bernice.
Hi.
Do you want to introduce yourself and tell us a little bit about the lab that you work in?
Sure. My name is Bernice Lynn. I'm a fourth-year PhD student in the neuroscience program at the University of Montana. My lab studies the bioelectric properties of nerve stem cells and how they influence brain development. We use Drosophila melanogaster as a model organism because they are evolutionary conserved, have a ton of genetic tools, short lifespans, and it's just a great model.
Yeah, that's cool. That's the fly.
That's the fly.
Yeah. Okay. All right. Well, let's maybe take a look at some of the stuff that you work with.
Yeah, for sure.
There's so much stuff here.
There's so many flies. If we ever have an earthquake, I'm huddling under this bench with all my babies. So this right here is a CO2 pad. This, because we work with flies, they do unfortunately fly away, and so the CO2 pad will put them to sleep.
Okay.
So I can do something like this little CO2 gun. And if you look at the vial, there are these flying flies, and if I spray CO2, then they start to fall asleep.
Oh, that's so cool.
Then I can put them on this pad and start working with them. And flies, like I said earlier, have a lot of genetic tools, and they have a lot of phenotypes that we can use to determine what genotype they have. So this example, if I can make it work on the camera.
And what kind of microscope or what kind of device is this?
This is just a regular old dissecting microscope.
Okay. So not a high power or anything like that?
No.
Not like an electron dot microscope?
No, or a confocal.
Which is huge, yeah.
Well, unfortunately, I am not able to get this camera to work.
I think that's pretty common, isn't it, with some of this tech?
Yeah, well, dang it. I should have played around with this more. But here are the flies, and you can kind of see that. And some of these, so this one in particular, they have curly wings. And that's just a chromosomal balancer. It's just, again, another genetic tool that we can use.
Okay.
So that's what the flies look like.
That's pretty awesome. I imagine a lot of your job is also troubleshooting equipment.
Absolutely.
Yeah.
That is, as you can see, this is my fly morgue. These are all my dead flies.
That's so sad.
It is very sad. So because we study brain development, we do a lot of brain dissections. I guess because that's not working right now, I will not show you that. But we do a lot of brain dissections. And I will show you some images of some brains.
Okay.
So right now I'm going to show you this software called Imaris.
Okay.
It is a roughly like $20,000 software, which is pretty crazy, but we can look at things in 3D. And so let me just show you some stuff.
There are probably so many different applications too between different labs.
Absolutely, yeah. So because this software is so expensive, I believe we're the only people on campus who have it, but we allow people to use it as well.
Oh wow! Yeah.
So let me just make this look a little bit better.
It's intense.
So essentially what you're looking at is a brain lobe of a fruit fly.
Mm-hmm.
And so in this program, we can turn it around, we can look at it, we can quantify it, it can count spots for us. So like if we wanted to count the number of these cells, we could do that, or the number of cells within a specific cell type or whatever. And that's really cool.
Are the different colors different?
Yeah, so
cell bodies?
Oh yeah, so let me just go slide by slide. So this green one, these are the what's called the type 2 neuroblasts of Drosophila. These are the neural stem cells.
Yeah.
There's only 8. It might be hard to see here, but there are 8 within a brain lobe in Drosophila, and it's a model that we could use to, it's evolution conserved, so it mimics human cortex development. And so that's what, it's really cool, and we can study these things in vivo, so within an intact brain, which is really cool. And then, yeah, we have all these different markers, and we can stain them with different things. Here is a whole brain. This is a larval brain.
Oh, yeah.
And that's really cool. And if I wanted to quantify the brain volume, I could also do that. And so by just using this, I can see what how this compares to other brains. And I can use the genetics of Drosophila to knock down certain proteins and see what effect it has.
That's so cool. And a lot of those effects are replicated in other organisms as well.
Exactly. It's evolution conserved. So even though they're a fly, the basic biology is the same.
Right.
And what's also really cool about flies is that they have reduced genetic redundancy. So one protein that I really look at is this calcium pump. In humans, there's 3 of them, but in flies, there's only 1. And with that, it has that reduced genetic redundancy, and I can, it's just more straightforward.
I see. Yeah.
Yeah.
More simplistic.
Exactly.
Yeah. That's so cool.
Yeah.
I really like that. So this was the Mars, and that was the Mars app, right? Right, okay. And you take the imagery from here and bring it here?
Yeah, so I can take you down to the confocal, and I can show you
sure, yeah.
kind of what that looks like.
Okay.
Let me just see if we can stop it.
Okay, so this is the confocal.
This is the confocal.
And it's so high powered, it really needs to be, that's why it has to be dark in here, too?
It does, yeah.
Yeah.
It uses lasers. So you get really high resolution.
Like IR?
IR meaning infrared?
Yeah.
Some microscopes can. It's just like another attachment. I don't think this one has infrared.
So here's where you would take the samples.
Yeah, so I would be normally dissecting the brains, and I would go through this process called immunohistochemistry, and that essentially stains the proteins that I want with an antibody, and then a second antibody with a fluorescence, so that we can detect it with a microscope like this.
You can't just put it under there without doing anything and see what you want to see. You have to separate it.
So sometimes you can. So you can have flies that are endogenously tagged. The proteins are endogenously tagged with like a GFP or like a green fluorescent protein. You can just dissect those and image them.
Okay.
Yeah.
Interesting. how many confocal microscopes does the university have?
So this university has this one, which is in the core facility. There is one other one that's really old and no one really uses, and then the Servan lab has their own.
Okay.
So basically 2.
Yeah. So not a whole lot.
Not a whole lot. Yeah.
And that's the reason why you have to reserve it, I would imagine, right? It's because so many people need it. Yeah.
Use it. Although our lab mostly uses it the most. So right now you're looking at a larval brain, and I used one of those endogenously tagged proteins. So this is just driven through genetics, and you can see these specific cell types. And then you can go through these different stacks, and you can take slice by slice, and then you can compile a 3D image.
Oh wow, okay.
And I can also show you, let's see, some other things.
Kind of like an MRI machine, how it does it slice by slice?
In that sense, yes, but it doesn't use like magnetism. It uses,
Right.
yeah, just laser scanning.
Right.
But if I wanted to make a Z-stack, so here I could just go set first, and then if I go up a little bit, and so here you're seeing it image it, all the lasers stack by stack.
So generally, how long does it take?
Yeah, so it depends on what you're imaging, how large your stack is, how many stacks. For my purposes, if I want a really high resolution section, it could be, and it's a relatively large stack, maybe 20 minutes for a single brain, or it could be as fast as like 5 minutes. And so there's a lot of parameters that you have to play around with to get the best signal to noise ratio and whatnot.
I see.
And then with these images, I can go into that software and then start to analyze them in whichever way I want.
Okay.
And that's pretty much what I do.
That's pretty cool.
And if I just go through this, you can kind of see all the different stacks. And in the blue, which I don't know if you can see very well, let's see, is all the DNA.
Oh.
And then the green are the neural stem cells.
Right.
And with just the blue, I can determine the volume of the brain. So we look at brain size a lot, and so I can quantitatively say if a brain size decreased or increased with whatever manipulations I did. Yeah.
Oh, that's so cool. It's so interesting seeing the DNA and the blastoma there.
Yeah. Super cool. Oh, didn't devote it too much. And this is only a 20x objective. I often will go into a 63x objective and just focus on a few cells.
Okay. That's impressive.
Yep.
So is that it?
That's it.
Well, I appreciate you
Of
doing
course.
that, Bernice, and
Of course.
it was really awesome to see all that and see it all put together that way. Yeah, we look forward to working with you again sometime.
Yeah, of course, no problem.
Okay.
Yeah, and if Katie's like, oh,